Hello, everyone, and welcome to this audio podcast. This audio podcast as part of Quantum Edition. This audio podcast as part of a Labostix sequence. This audio podcast as part of a Vortex Gauntlet, and this audio podcast as part of the abstracts, and this audio podcast as part of a Storm Labostix production. In today's, as part of today's audio podcast, I look at an abstract as part of works from 2015, looking at nephrotoxic nephritis and glomerulonephritis, animal model versus human disease. The works formed a publication in the British Journal of Biomedical Sciences. In around 2014, 2015. So the abstract as follows: glomerulonephritis, or GN, encompasses a range of immune-mediated disorders that cause inflammation within the glomerulus of the kidney. The pathogenesis of GN glomerulonephritis is complex, and intricacy arises from factors such as autoimmunity, cancer, structural abnormalities within the kidney. Studies using laboratory animal models have highlighted crucial interaction between inflammatory cells and cells intrinsic to the kidney, both of which are fundamental to the pathogenesis of GN. As part of the sort of as part of the aims of this works was to provide insight on a suitable model for nephrotoxic nephritis and glomerulonephritis, or NTN and GN, and relate experimental validity to humans. The BALBC NTN murine or mice laboratory model, and Wistar Kyoto or WKY laboratory rat have held experimental validity in the study of GN in humans. The the chemokine receptor CXCR3 also mediates renal T cell recruitment and subsequent tissue injury in a nephrotoxic nephritis. It is noteworthy to consider CXCR3 blockade in TH1 mediated renal inflammation as prospective therapeutic options for patients with GN and subsets thereof. Currently used immunosuppression of pharmacotherapies for GN are not always uniformly effective and are frequently associated with side effects. Corticosteroids are effective in several types of GN, owing to ability to prevent pro-inflammatory effects of cytokines known to promote glomerular inflammation. Differences between experimental and human GN complicate translation of experimental therapies into practice. More research is required to translate animal model research into a better comprehension of human GN disease. However, the complexity of GN research makes findings a challenge to replicate. Some terminologies that can be identified some terminologies that can be utilized to identify similar works include chemokines CCL2, glomerulonephritis, macrophage colony stimulating factor, and nephrotoxic nephritis. That draws this audio podcast to a close. This audio podcast as part of a quantum edition. This audio podcast as part of a Labostix sequence. This audio podcast as part of a Vortex gauntlet. And this audio podcast as part of the abstracts.